So the point of this picture is that there's a lot of variants around the world, and there's a couple dominant variants we all know about, alpha, beta, gamma, delta, but there are many more other than that. They just don't really make the news. And there's a lot of green on that map, and that's, as you probably guessed, is delta, right? Delta is very contagious, and it's spread all around the world. And this second plot is a frequency plot that shows all the variants over time up until now. So their alpha was kind of a big deal for a while, but that got displaced by delta. But I want to draw your attention to the red line, right? That red line starts all the way back at alpha, but it's kind of creeping along and building, and that's the Omicron strain, which has been found to be basically most related to alpha, which is kind of interesting because you might think that over time the mutations are sort of linear, right? Like alpha might lead to delta, and delta might lead to Omicron, but that's not exactly the case. There's a lot of diversity in the viral genome, which can be shown by a phylogenetic tree like this. This is a little bit out of date, but I'm just going to introduce it now and talk about it a little bit later. But I do want to mention that there was a recent paper from August of this year that was done published by the University of Bath and the University of Edinburgh, and they found, or what they're suggesting, is that the true SARS-CoV-2 mutation rate is at least 50% higher than what was previously thought, which is a lot. They were kind of, I guess, concluding from that, if the SARS-CoV-2 infection passed around on average with this mutation rate, that there'd be about 300,000 new mutations passed from one host to another every day, which is a monumental amount. Obviously, there are not that many strains, so why is that? Well, not all the mutations are beneficial. Most of them are deleterious to the virus, but a few obviously give it an advantage. I'm going to point out this other paper that was published just a few days ago, December 1st, that was done in this state, Washington, by the Washington State Department of Health. And they're basically tracking breakthrough cases of SARS-CoV-2 variants, and it was all of last year, or basically all of this year. And by breakthrough cases, they mean that individuals have been vaccinated, they've had their full vaccine regimen, but they still acquired the infection all the same and became ill, which is not good. And breakthrough cases are caused by largely variants. I mean, you could be immunocompromised, but variants are a big problem. And so what they found is that there was 78,000 breakthrough cases in Washington this year, and about 775 people died, which with respect to the population isn't a huge amount, but it's more than it should have been. One final thing about this paper I thought was interesting. Over 50% of the breakthrough cases were the age range 20 through 49 years old, which I'm pretty sure encompasses everybody in this room. So it does apply to our age group.
So why do these mutations matter in the context of eliminating the burden of COVID-19, specifically in terms of testing and vaccination? So briefly, the SARS-CoV-2 viral entry mechanism involves the receptor binding domain of the virus attaching to ACE2 receptor, which is present on human cells. And so these variants have shown to increase transmission, for example, Omicron being twice as likely to be passed on, and as well as Delta increasing transmission and infectivity. So vaccines against the virus are important because they decrease the likelihood of severe disease. And so as an example, if we are given an mRNA vaccine that encodes for the spike protein, it allows our bodies to make the spike protein so that we can naturally make antibodies to recognize the virus. These antibodies can stop the virus from replicating if we were to be infected. However, when SARS-CoV-2 mutates, then the antibodies we developed might no longer recognize and bind and neutralize the virus since it has changed. And so decreased vaccine efficacy means increase in deaths, increase in spread, and higher likelihood of variants developing. And so currently there are a variety of methods to detect the virus. Antigen tests, for example, detect proteins on the surface of the virus. And this can be accomplished by taking either a nasal or throat swab and adding it to a lateral flow assay type test. However, these tests do not specifically detect for various mutations, and frequent mutations might make these tests obsolete, and they are less reliable due to low sensitivity. typically require confirmation from another test. However, they do provide good specificity, and they are pretty rapid.
So I'm going to talk about some of the limitations and just kind of the current state of the art for nucleic acid amplification tests. So we've already heard about these tests. They're highly sensitive. They're resistant to false negatives. As Kevin said last week, PCR is the gold standard, but it does require a lab and a thermocycler and likely a technician also to run it all. NATs have good specificity, and you can also do an isothermal amplification with them, where you don't need a thermocycler. And it does have great performance, but on point-of-care devices, it can lead to low sensitivity. So what's lacking in the mutation detection space? Well, currently, as far as we know, there are no point-of-care devices that are specifically designed to keep up with the variants. But this poses a problem of a risk of false negatives if you have a variant that's not being tested for. So current variant tracking is done in laboratories using sequencing technology, and they get their samples from their excess patient samples that are donated for screening. But we're always going to be 2 steps behind of the virus if we approach detecting variants this way. And currently, there are really no point-of-care devices that are dedicated to tracking and anticipating future mutations of SARS-CoV-2. So basically, what we want you to take away is antigen tests and nucleic acid amplification tests for point-of-care diagnostics have advantages and weaknesses. But what if we can combine both of these tools and leverage their advantages to build a better variant tracking diagnostic? So we came up with a need from all this information that there is a need to develop a point-of-care diagnostic that can be disseminated broadly with the functionality to test and anticipate the SARS-CoV-2 mutations in the viral genome and to track the structural and functional changes in the receptor binding spike protein in order to identify and characterize mutations with strong receptor binding or mutations that evade the immune system.
And so we hypothesize that by creating a dual antigen nucleic acid detection test, we can rapidly and with high specificity detect various SARS-CoV-2 mutants. And our device will decrease the transmission of COVID through frequent testing, provide a better understanding of the implications between mutations across the SARS-CoV-2 genome and phenotypic impacts, and allow for improved vaccine efficacy through predictive mutations and allow for future vaccine development. So in the first aim, we will be building an antigen test that can recognize different variants or possibly predict the occurrence of new variants. And so we will accomplish this through a split protein recognition immunoassay. So for example, HRP is an enzyme that oxidizes a small molecule. Upon oxidation, the small molecule turns blue. And so HRP has been split so that it is non-functional at room temperature. However, if you genetically attach 2 proteins onto each half that recognize a common target, then the 2 halves are now in close enough proximity to bind and become functional again. And so in the context of testing for SARS-CoV-2, we can genetically fuse antibodies that bind different domains of SARS-CoV-2 receptor binding domain, which has been seen as like the most likely area for mutations. And these antibodies will be modeled after the antibodies are bodies produced from vaccination and natural infection. And so we can create a library of different split enzymes genetically fused to antibodies that recognize the various variants. And then we can also add an ACE2 capture protein that will be included following the variant antibodies. And so if it binds here but not to the previous antibodies, then this might indicate the possibility of a new variant. And this will be a useful tool to not only keep track of the variants, but avoid having to wait weeks before a new variant is detected.
So for our second specific aim, it's focused on the nucleic acid amplification test. And really what we want to do is build a massive mutation library and put it on the device. I'm just going to say we assume that this is going to be an isothermal amplification. We could use LAMP rolling circle amplification or any of the others. But there is one constraint. It has to play nice with the antigen test, and that we're just going to assume that it's going to be done at room temperature, such that we don't denature any of the proteins on that side. But how do we predict the sequences to look for? Well, there's two strategies that we can apply. One is a genetic algorithm. Well, both of these strategies can be combined, really. And the second is the selection rules for the genetic algorithm. So what is a genetic algorithm? It's an algorithm that's used in computer science, but it could be used in many things. You basically take an initial, whatever you start with. In this case, it's a strain, alpha, delta, or omicron. And then you take it and you mutate it. You perturb it a little bit, and you produce a generation from that. Then you apply selection constraints on that second generation to weed out a bunch of the mutations that are unfit and that would likely not persist on. So you select away all of the weaker mutations, and then you're left with a subset, only a handful of that generation left. And you can keep doing this. You can perturb that generation, producing even more. You can keep doing this for a third generation, and you can do it as long as you want. But the point is we're trying to basically recreate the phylogenetic tree of the virus, right? And we can never do this perfectly, but this is kind of like one of the better ways that we could approach it. And this is an up-to-date phylogenetic tree. You can see deltas just all over the place. It's massive. So what are the rules for selection? There's a couple that we could use. One is called trinucleotide base pair stacking free energy analysis, which is a mouthful. But basically what we would want to do is look at the genome. I realize this is DNA and not RNA, but what we would want to do is look at the genome and look at three base pairs and basically mutate the base in the middle and see, does the energy of these three decrease, or does it become more favorable or less favorable? And you can score based on which base you're replacing with. And you can keep doing this, keep shifting your frame all the way through and apply this analysis every single time. You can come up with a big scoring scheme and possibly a Pareto chart, and you can basically take the best 1,000 or 500 or however many you want to keep from this system. So there's a couple other strategies, and these actually have more promise in my opinion. This was found by the people who submitted that paper in the beginning from University of Bath and New Edinburgh. They found that the virus SARS-CoV-2 has a high propensity of mutating guanine to uracil and cytosine to uracil. And these mutation rates are substantially higher than all the other mutation rates, especially as you start adding up the mutations. So there's obviously some selective pressure there. There's also selective pressure for incorporating stable amino acids, and from the virus's point of view, this makes sense because if there were unstable amino acids and they fell apart, it would take more energy and more effort for the virus to replace it and fix it. So this is inherently a selective pressure, and that's what they found. And then lastly, they've also found that mutations that conserve amino acid types were very, very common. And so what we can do is apply all of these rules essentially to our selection algorithm to really weed down what we think are the very best mutations that will persist on. And then as for the detection, we cannot do a colorimetry or fluorescence detection, or because that would interfere with the other part of the assay. And there's just too many targets, like 2,500 or something, that we would put on the test. So what we would have to do is a direct detection method. And there's a number of ways you can do this, but the simplest way is if you have two electrodes, you can conjugate a single-stranded piece of DNA across them. And upon a hybridization event, it can change the conductivity of that circuit. And this is really easy to record. You can make a bunch of nano circuits or wires, a whole array of them to do this kind of testing. And this is totally independent of the antigen test. I guess I'll do the slide. We didn't decide. So in conclusion, what we're proposing is multiple tests on one device to provide protection against false positives and false negatives. The pros of the antigen test, they're rapid. The pseudo-testing antibody neutralization and Immunoassay, I'm not sure.
Oh, yeah. And they kind of provide us way to test if the antibodies we're producing from vaccination or just from natural previous infection are escaping or if the new virus is escaping that neutralization, and it helps with the detection of possible new mutants.
And the pros of it should be NAT, not NAC. Testing is that it's rapid, sensitive. It's good, and it's a good screening method for mutations in the viral genome, especially when you're trying to look at maybe many thousands of potential mutations. And the takeaway is that this, yes, is a blue sky type of device, and that would be very high cost, at least initially. But ultimately, this will probably be lower in cost than producing ineffective and endless vaccines and treatments. And the idea is that we become, up to speed and toe to toe with the virus as it's mutating, so we can better position ourselves against it. Thank you.
Fantastic. Unfortunately, we don't have time for questions, but you can ask any questions on your last discussion board assignment.
Thank you guys.
That was fantastic. Sorry.